So 11.4 is meiosis. And meiosis is a reduction division process used to create the gametes or sex cells in the human body. Homologous, meaning that each of the 4 chromosomes that came from the male parent has a corresponding chromosome from the female parent. So what we're looking at here is homologous, meaning that you have half the genetic information on one, half on the other, and those homologous chromosomes are what's going to be separated during meiosis. Now, diploid means 2 sets. It is a cell that contains both sets of homologous chromosomes. The number of chromosomes in a diploid cell is sometimes represented by the symbol 2n. Right? So diploid means both sets of homologous chromosomes. If you separate them out, you'll have allele on one, allele on the other, and then they can be passed on to the offspring as just N. Right? The haploid cells will be N, which is what we see right here. Haploid is a single set of chromosomes, so it's only got half of the genetic information, one of the alleles. And meiosis is a process of reduction division in which the number of chromosomes per cell is cut in half through the separation of homologous chromosomes in a diploid cell into a haploid cell. So you take the full amount of genetic information, you cut it in half. Now a tetrad is when the homologous chromosomes pair with each other during prophase 1, and they cross over, they become recombinant, and they exchange a portion of their chromatids. All right, a portion of the genetic information is passed between homologous chromosomes, allowing for variation between the two. So if we look at this pretty blurry diagram, we can see that in prophase 1, which is the second bubble from the left, we see the yellow and the red X forming a tetrad, and the blue and the green X forming a tetrad, and they undergo crossing over. And if you look in metaphase 1, which is the middle one, where they're lined up along the center, you can see a little bit of yellow on the red and a little bit of red on the yellow X. Now in anaphase, we are separating the homologous chromosomes, anaphase 1. In mitosis, we separate the sister chromatids. In this case, the sister chromatids are still together. We separated the homologous chromosomes. Now if we look at mitosis 2, now we are still going to have metaphase, anaphase, telophase, and cytokinesis, but we have these sister chromatids separated. And that's how we end up with 4 haploid cells, because we start off with doubled genetic information. So in this first picture here, interphase, we've doubled. In prophase 1, they are 4N. And at the end of mitosis 1, they are 2N. And then at the end of here, they're just N. Mitosis is a production of 2 genetically identical diploid cells, whereas meiosis produces 4 genetically different haploid cells. And if we take a look at this diagram right here, we can see the difference between male and female meiosis. Males form the sperm, females form the egg. They only form 1 egg and 3 polar bodies, though. We'll quickly just move through section 5. In linkage and gene maps, it is the chromosomes that assort independently, not the individual genes. And gene map shows the relative location of a gene on the chromosome. Okay, so if we take a look right here, here's a fruit fly gene map, and that's what they were talking about in that one video about the Human Genome Project, where they've mapped out the locations on the chromosomes.